we'll first learn to prepare data into count matrix from the output you learned from Josh's last lecture. And then we'll introduce the basics for the seq and the enrichment, gene set enrichment analysis. We'll also learn to implement both in R. So first, now that you have been to Josh's last lecture, what you are having for the DE-seq experiment are these output files that contain the read count. Each file corresponds to one run of one sample. There are 4 runs for each experiment. So we're going to combine all those individual runs into the same count matrix, and that will be used for the DE-seq object and also the other downstream analysis. This line of code is trying to get all of the file names that end with this format. These contain the read count for each run. So here are some of the examples. And we're going, in total, there are 96 files for 24 experiments. Each experiment has 4 runs. We're going to combine all of these files into a count matrix that is 24 by the number of genes. So if you look at each individual file, this is a file name of one of them. We're going to take 2 of them as example, read them in using these code. This read TSV file will let you read in that each individual count file. And then you can take a look at what is inside. It is this number of rows multiplied by 4. So that each row here, if you look at inside, each row corresponds to 1 gene. But the first 4 rows are the number of unmapped reads, multi-mapping reads, no feature reads, and ambiguous reads. You should have learned the difference between those in the previous lecture. And the 4 columns, the first one is the gene name. The second one is for unstranded library prep. The third is for the library prep that is for the coding strand, and the last column is for the antisense strand. So depending on how your library is prepped, you will use 1 column of them, depending on how your library is prepped. In this specific experiment, we're going to use the last one. So I think to me, if you don't have a clear, like if you don't remember how your library is prepped, I can tell from this column that the last one is the one, because this one has a large number of no feature reads. So from what you learned from the last lecture, this is actually the summation of all the other reads, right? And this one has too many ambiguous reads because it's checking double strand. So that's how I can vaguely tell that the last column is the one that I should use for summing up the read count. And then we are going to use this select function to get the 4th column, and we'll do this for each individual file and combine them. And in order to combine them, we'll need to first rename this column, because this is for 1 experiment, so for 1 run. Each run will have a column that's X4, and if you combine them, you need to rename them by the run name. So I'm renaming them by the file name, and then I'm going to combine them by using this full join function. So this is taking the 1st column and then combine the 2 files, two runs into the same count table. Now I have 2 columns of the count. And because we have 96 count files to combine, we are going to run through a for loop to combine all of them. There are multiple ways to do this, and if you're very proficient in R, you can definitely write your own codes to do this. But here we have a set of some codes that make it really efficient for you guys, so you can immediately borrow them to combine all of the 96 files. So I think this is repeat.
That code does not use a for loop.
Not here yet. So this one is the one that with the for loop. There's a for each. This is a very powerful function in the package for each, named for each. You can also write your own for loop. This is just a syntax that's very convenient to use, but the concept is the same. You are going to first, for each of the file, you read in the file, you get the file name, you read in the file, you select the last column and rename the gene by the file name, and then you will join it with the previously read file, and you will do this for a loop. So this will merge file 1 and file 2, and then merge file 3 to the first 2, merge file 4 to the first 3. This whole for loop is going to do this. Does that make sense? So eventually you are going to end up with 97 columns with the first column for the gene name. It will look like this. Each column is one run. And remember, we have 4 runs for each experiment, and we will combine the 4 runs, we'll add them up to obtain just 1 column for each experiment. In order to do that, we need to know which columns correspond to the same sample, same experiment. So to do that, we'll link the count matrix to the metadata. This metadata is recorded in this CSV file on the GitLab repository. You can download yourself. And the content of this metadata table is you have this is part of the file name of the read count. This is each run's name. And this is each experiment name. And you can see each experiment has 4 runs. So we're going to get this information, map this information into this count table so that we can group the 4 runs from the same experiment. So here, this is trying to get the file name. This is the common part of the file name. We're going to remove this by replacing this by nothing. And so you're left with the key part of the file name. And this is called file name short in this new data frame. It will have the file names and the short name of the file names. And then we'll also add another column in this data frame called library. So this will take this data frame's first column. It will take the column called experiment and match each row to the file name. So after doing this, you will have a data frame that has the experiment number mapped to the run file names, and you will be able to sum up all of the 4 runs. So that's done using these codes where you have, you will remove the gene name from this whole data matrix because you're trying to sum up the counts. The gene name is 1 column that's not useful here. And then you will, this is the key part, you will join the, join this data frame that contains the library, mapping between the library and file name. You will join this by with the with the count matrix. So, and then you will have more rows. Originally, there are I think there are two fewer rows, and now you have two more rows representing the file name and libraries. And then with this one, these are the two extra rows. Two extra columns. And then you will be able to group them. The next commands are going to sum up all of these 4 rows into 1 row. So here, this is going to do the summation. Group by library, summarize them by sum, and then clean up the whole operation. So I think I've just been copying using these codes. If you're not very familiar with these packages, then it might read a little alien to you. But what this achieves is to sum up the 4 rows sharing the same library. And then eventually we want it to be recorded as a matrix. Because sometimes DSeq doesn't like data frame as the input for the count. It likes the input as a count matrix, not a data frame. And this is the count matrix we have ready for DSeq. It will have genes on the rows and the samples on the column. And then we'll also process the metadata frame so that we only have the experiment, not the samples anymore. So this is going to be a 24 row data frame instead of 96 rows. It will tell us for each experiment what is the genotype of the mice, what is the median use for that treatment. And for this specific experiment, the gene names, some of them have this string inside the gene names. They are, I believe they are the non-coding genes. So sometimes you would need to remove the genes that you are not interested in at all in this step. And you can also exclude the low expressed genes. So here, this is trying to remove genes that have read count this is trying to keep the genes that are expressed in at least, that has at least 10 reads in at least 3 samples. So you can change these numbers depending on your experiment. Sometimes when you have very dramatic difference between 2 treatment groups, maybe one gene is expressed in one group and not expressed in the other group, you may want to do the filtering by group, filtering of the genes by group instead of just treating everyone at the same time. Here, I'm just doing a very commonly used threshold for the filtering of the lowly expressed genes. And some people do not do this step before DSeq. It really depends on what your goal is, but I personally like to filter some of the low expressed genes. You may loosen the criteria a little bit, but it's still helpful to remove the really low expressed genes, because first, by including those genes, your DSeq will have to adjust for more genes in the multiple comparison. So it affects your power. And most of the times, these lowly expressed genes are not differentially expressed. And by filtering these genes, you will also save some hard drive space in your saved object. And also, if you try to do some quality assessment, having these really lowly expressed genes may not be informative in the quality assessment. So after this step, we'll have a count matrix for the genes and the metadata matrix data frame for the samples, and we'll save each of them in a CSV file ready for use in downstream analysis. Any questions? So after you get these counts, a very commonly used quality assessment is to plot a principal component analysis of the samples to see if there are any obvious batch effects, or if your treatment effect is super strong, can you already visualize that in the PCA plot? So for example, here, we're going to plot them colored by the genotype and shaped by the media. So this is telling us that there's a strong effect in both variables in this specific data set. And sometimes you can color them by batch and see if the batch separates a lot, or if you have like very obvious outlier in the data set. So in the implementation, when you do the PCA plot of the count, remember that the concept of PCA, principal component analysis, is to look for the direction of maximum variation. And count data, especially RNA-seq data, their variance is generally dependent on the mean. So the higher your expression level is, the more variance you can have. And to do PCA analysis on these kind of count data, you need to stabilize the variance such that the variance is almost constant for the mean. You don't want like your cluster of points to be like very tight for the low expressed genes and very spread out for the high expressed genes. So in order to do that, there are two transformations on the count data before PCA. One is the VST transformation, variance stabilizing transformation, and the other is the R log. This is doing just the log transformation of the counts with one plus inside. So both can stabilize the variance to some extent, and make sure you use the transformed ones, the transformed counts before PCA. Questions? Yeah.
So you would only apply either the R log or the PCA.
Yeah, we don't have, yeah.
And it's kind of user choice.
Yeah. So that's the preparation of the data. Now that we have the count matrix, we can move on to the main content of today, the differential expression analysis. So there are multiple R packages used for differential expression, and you probably see them a lot: DSeq, EdgeR, and Lima-Voom. DSeq and EdgeR, they model the count directly. Lima-Voom, it will transform the count data into some continuous data and model the continuous data. There are benefits on each side, pros and cons on each side. If you model the count data directly, then the model itself will account for the different sequencing depths itself. You don't have to normalize that size factor out. By size factor, I mean the sequencing depth, like different libraries will have different total number of reads. That will be automatically modeled by the count models, such as DSeq and EdgeR. And it can handle zero count better because you're not doing any log transformation or anything. You're modeling the count, so zeros can be treated as is. But for those count-based models, they are less flexible to more complex model design of the experiment. For example, if you have repeated measurements and if you want to include some random effect, then DSeq and HR cannot include that. But continuous data-based model will be able to handle that because it's essentially transforming the count data into continuous data and model it using linear models. And linear regression can have so much flexibility, including random effect and all kinds of stuff. So if you have a very simple model design, then DSeq would be a good choice. But if you have a more complicated design, then maybe you should think about Imago. For this lecture, we're going to focus on DSeq. So DSeq models the count data. How? There are distributions for continuous data, and Gaussian distribution is one of them. For count data, there are 3 very commonly used distributions. The first one is Poisson distribution. And the Poisson distribution is often used to model the count of occurrence of events. For example, if you're in a bank and you're trying to count the number of visitors to that bank in a certain period of time, that number of customers is going to follow a Poisson distribution. And the Poisson distribution has a parameter that's the mean parameter, which is basically the length of time you're trying to count the number of customers coming into the bank. If you're counting within 1 hour, that's one Poisson distribution. If you're counting by 2 hours, that's another Poisson distribution, and the second one will have a larger mean. So that mean parameter will tell you how much the average count is going to be. And here are 3 examples of Poisson distribution with 3 different mean parameters. You can see their peak number is going to be different for different mean parameters. So Poisson distribution is very intuitive, but it has a drawback. The mean and the variance are exactly the same. If we use Poisson distribution to model the recount, then we're assuming that highly expressed genes also have high variation. But the relationship between mean and variance for genes are not that linear. So there are genes that are highly expressed, both highly expressed, but some may be more, some may have a more high variation, and some may have a lower variation. You don't want a distribution to model the count data such that the mean and the variance are completely tied together. So that's why Poisson distribution is not used for RNA-seq data. And the second very popular count distribution is binomial distribution. This is the one that you guys all have seen a lot in undergraduate classes, flipping a coin, calculating the probability of getting 3 heads and 1 tail. So this distribution is also if you look at, so this distribution has 2 parameters. One is a fixed parameter, which is the number of coin flips you do. And the second parameter is unknown, which is the probability of getting a tail or a head. So those 2 numbers will determine the probability of you getting a head or a tail. So here I have one example. Instead of flipping coins, we are going to have a board that's 40% white and 60% black, and you randomly drop 4 beans onto that board. So what is the probability of having 0 beans on the white and 4 beans on the black part? That is going to be the area for the black to the power of 4. If you have 1 bean on the white, 3 on the black, then this is the probability for the 1 bean on the white. This is 3 beans on the black part, and you need to choose among 4 of the beans which one is landing on the white. And similarly, you can calculate the probability of each event happening. That gives you this probability function that describes the whole binomial distribution. And you can calculate the mean and variance of this binomial distribution. They are still tied together. So even though you have two parameters, this n, this total number of coin flips or total number of beans, this is already given. The only free parameter is p. So your mean and your variance are still going to be completely tied together. And that's why we will not use this for RNA-seq data as well. But those two distributions are very important in the understanding of why we use negative binomial distribution for RNA-seq data, because negative binomial distribution will have the freedom between the mean and the variance parameter. It will allow us to model the variance as something multiplied by the mean, and that something is called the overdispersion. It's like the extra variance on top of Poisson binomial distribution. So negative binomial distribution is a little harder to understand intuitively. So it's one example is, suppose I really want a girl, and I keep giving birth to kids until I have a girl, and the number of kids before I have one girl or two girls, that is going to be a negative binomial distribution. And the whole function to describe the probability of each event happening is here. And based on this function, you will be able to calculate the mean and the variance. So this P here is the probability of having a girl. And this R here is the number of girls I have before I stop giving birth. So the mean parameter is these contains these two free parameters, and the variance parameter can be written as the mean multiplied by something. This something is larger than 1. And this is the overdispersion parameter. It means that my variance can be larger than the mean, and this by how much larger is determined by a free parameter. And if you are interested, this negative binomial distribution can be reparameterized. So if you look at different Wikipedia pages or people explaining the concept of negative binomial distribution, you may see one form instead of the other form. This second form is going to be used for the estimation of the parameters, because it has this mean parameter and overdispersion parameter here. This is like easier for your intuition instead of the original parameterization, the P and R. This is the probability of having a girl. This is the probability of having this is the number of girls I have before I stop giving birth to. So if you think about that in the context of RNA-seq data, it doesn't make sense. We're just using that distribution to describe count. We need a distribution that has different specification for mean and variance. So that's why we parameterize it into here. And this one will describe the distribution with mean and extra variance. And so the larger the overdispersion parameter is, the heavier tail you will have on the distribution function. So here I have the purple as the Poisson distribution. It has the thinnest tail if you look at the bottom here. As I increase the overdispersion parameter in the negative binomial distribution, I'm changing from blue to green to red. You can see the tail get heavier. So the RNA-seq data generally have a fatter tail than the Poisson distribution. That's why we use the negative binomial distribution to describe RNA-seq data. And for each gene, you may have a different spread, different overdispersion. So each gene will actually have a different overdispersion parameter that's going to be estimated. Questions? So how do we estimate these parameters? Most of you guys probably won't really worry about it, but if you are curious, the philosophy behind the estimation of the parameters is something called the maximum likelihood estimate, MLE. How many of you have heard about that before? Okay, so suppose that we have a coin flip as an example, and we flip 10 coins and got 9 heads. How would you estimate the coin's probability of getting a head? So 9 out of 10 are heads. How do you estimate the probability of getting a head? If you estimate it by 0.5, if you think the coin is a fair coin, then the probability of observing 9 heads out of 10 is this number. If you assume the probability of getting a head is 0.9, very biased, then the probability of getting 9 out of 10 are heads is this number. And you can see that this probability is much larger than this. That means this guess is a better guess than this guess. And you will do this guess for all possible values of P. Each P, you will get a probability of observing your current event, and you will choose the parameter value that gives you the best probability of observing the current event. So that's the philosophy of MLE that will be used for the estimation of the seq parameters and also linear regression, logistic regression, all the things that we talked about in the last part of the workshop. So for negative binomial distribution, we're going to use that to model each gene. And if you only have one gene, then you can directly apply the MLE method to this likelihood, which is the probability of observing all of your gene counts for this specific gene. For all of the samples, you will have a likelihood, the probability of observing these counts. And you will maximize this probability to obtain the estimate for each coefficient. This is done for only one gene, but if you have thousands of genes, you will do this for thousands of genes simultaneously. And in this simultaneous, we'll talk about how we do it simultaneously. But remember that in the seq, we are trying to link the recount to some experimental conditions like genotype or treatment. How are we going to link the count to those conditions? That's through the parameters in the negative binomial distribution. So here, remember we have the mean parameter in the negative binomial distribution. We're going to model the mean parameter through the linear combination of the predictors. This is similar as logistic regression. Because the mean of the count is going to be a non-negative value. But your linear combination of the predictors, like genotype or conditions or age or whatever variables you have, this is going to be a free range variable. So how do we link this to a non-negative mean parameter? We're going to take 2 to the power of it. So this 2, instead of some other natural log or log 10, is just for the ease of interpretation. Because right now, if you increase, for example, x1 by 1 value, by 1 unit, you are going to increase log 2 of the mean by beta 1. That's going to multiply mu by 2 to the power of beta 1. So that's why these coefficients are also called the log 2 fold change. You're trying to model the fold change of mu by these coefficients. And this is the log 2 of the fold change. Does it make sense? This log2 function is also called the link function. It will link the mean parameter of the count to the predictors. It is in this shape. So you are going to have a non-negative value linked with a range value that's from the predictor. So one thing to remember that we're not linking the predictors directly to the counts. The counts themselves are random variables. They are not completely determined by the predictor. The predictors are determining the mean of the count. The count can be observed with some measurement error based on the mean. So we're not really modeling the count directly by the linear equation. So here I'm going to repeat again why the parameters are called log2 fold change. Suppose that we have two predictors, and these should be bullet points. We have one predictor for the condition and one predictor for the age of the samples. And the mean of the count will be modeled by this equation. And you can plug in, for example, for an untreated 40-year-old, the mean count, like this is the expected count of this person is this number. And if this person, instead of being untreated, is treated and is still 40-year-old, you will add, you will increase x1 by 1 because x1 is the indicator for treatment, x1 is 0 for untreated, x1 is 1 for treated. So the difference between the untreated 40-year-old and the treated 40-year-old will just be this beta 1 here. And so that's why the meaning of beta 1 is when age is unchanged, the expression is going to be multiplied by this number if this individual switch from untreated to treated. This is the expected fold change. So the interpretation of each parameter is given the other parameters unchanged, the fold change of this corresponding variable. And to get the meaning of beta 2, you're going to keep the treatment the same between two individuals and compare this treated individual that's 40 and treated 41-year-old. These two are only different by 1 year of age. Their treatments are the same. So the only difference is in the beta 2 here. This beta 2 is when condition is unchanged, the expression is expected to be multiplied by 2 to the power of beta 2 if age is increased by 1 unit. Everything needs to be interpreted in terms of all the other parameters, other predictors unchanged. And in this equation, the beta 0 and the over dispersion parameter, they will be estimated. They're important, but they are not going to be interpreted. So they're also called the nuisance parameter. The dispersion parameter here is very important. It will affect the length of your confidence intervals. The larger the dispersion is, the wider your confidence intervals will be. And this changing of coding variables, I think we've covered it in the previous part of the workshop, but it's a very commonly seen scenario, so I'm going to talk about this again. Most of the times when you have a categorical variable, for example, treated and untreated, you will choose one of them as the baseline level and code it as 0, and the other one code it as 1. And the category chosen as the reference level that's coded as 0, that one is up to your choice. So here we have originally coded the treatment group as 1 and the untreated group as the control group as 0. But you can do it the other way if you have two categories that are not clear which one is the reference level. And if you change the reference level, if you switch the 0 and 1 for the two groups, you're going to get the same result. It's just a flip of signs. How does it work? So suppose that this is the original coding. You will have 2 to the beta 0 for the untreated, 2 to the beta 0 plus beta 1 for the treated. The log fold change, beta 1 is the change when you switch from untreated to treated. But if you do it the other way, if you use 1 for untreated and 0 for the treated, you are essentially coding a new variable that's 1 minus the original 1. And if you rewrite the mean of the read count, you will have a different expression. And this is the new regression coefficient for your treatment variable. It will have a minus sign in front of it. So, and this new whole thing is the effect when you change from treated to untreated. So everything, the value, the fold change is going to be the same. It's just a flip of sign. And in terms of the fold change, it will be the inverse. Do you guys have any questions? And if you have more than 2 categories, there will be 2 variables coding for each of the non-control categories. This was also covered in the last part. So suppose I have 2 treatments and 1 control group. I will use 1 variable for each of the treatment groups in reference to the control condition. So this is going to be 1 if it's treated by A. This is going to be 1 if it's treated by B. And for the samples treated by the condition, the control condition, it will be 0 for both variables. So this is the mean expression for the treatment group A. This is for treatment group B, and this is for the control condition. And you can see beta 1 and beta 2 are each of the fold change. They are the fold change of treatment A and B in comparison to the control group. If you want to know about the fold change between A and B, you will need to take the difference between beta 1 and beta 2. Does that make sense? So the control condition here is the baseline. Most of the times, you may have just 1 category that's naturally used as control. But in some other situations, it may not be that clear. And in those situations, you may want to, the simplest way is to code the one you want to compare to as the baseline. If you want to do pairwise comparison, then it's more complicated. You can either use the DE-seq contrast to obtain the difference between A and B group, or you can rerun DE-seq with A and B as the control category. So if it's too hard for you to get the full change between A and B, you can use A as the reference level and rerun DE-seq again. So now we have covered the very basics building blocks for DE-seq. We covered the count distribution, how we obtain the parameters of the count distribution through MLE, how we link the predictors to the count, how to interpret the coefficients for each predictor. And now we're going to go to DE-seq, where we're going to model thousands of genes together. So each gene will follow its own negative binomial distribution. And in this negative binomial distribution, this mean parameter is going to be modeled by this linear equation of predictors multiplied by a size factor. This is slightly different from before. This size factor is going to account for the library size. So even if we're modeling thousands of genes, we have one model for each gene. Those genes has one thing in common. They come from the same library. So that size factor is going to be shared across the model of our gene. It's like you have one gene, you have one model, but each of those columns, they are connected by their size factor. We're weaving all of the thousands of negative binomial regressions together through the shared size factor. And this over-dispersion parameter is going to be gene-specific. The mean expression level is going to be determined by each gene's coefficient and each sample's predictor. So you can see the index I for gene, J for sample, both appeared in the mean expression. The J for sample only appeared for size, and the I for gene only appeared for the over-dispersion. This is like you're modeling a really big matrix using all these negative binomial distributions, and it will calculate very efficiently, fortunately, through DESeq. In order for DESeq to run, you just need two things as input: the count matrix that we obtained for that's the gene by sample count matrix, and then the meta matrix that tells you for each sample what are the values of the predictors. What predictors you are going to use is stored here. It is always important to check whether their row names, their sample names are matched together, because if they don't match, then DESeq may give you an error. And for DESeq package, it will automatically transform if you're trying to use a categorical variable as a predictor, it will automatically transform the character string into factor variables. But if your treatment variable is recorded as a numerical value, for example, it is 1, 2, 3, even if you know 1, 2, 3 are 3 different treatment groups, R doesn't know, it will treat it as a continuous value. So DESeq is also going to treat as continuous. So it's important for you to transform that as a factor variable before the input of DESeq. So here I'm going to transform the two predictors of interest as factor variables. And you can see in this data set, we have a 2x2 design, factorial design. There are half that are wild type, half of the mice are this genotype, half of them treated by this media, half are this media. So it's like 6 for each of the 2x2 combination. And the DESeq, before running DESeq, we need to determine what effects we're trying to estimate. So what is the design of what differential effect we're trying to estimate? That is stored in this design formula. So you can use, if you want to check the differential expression in terms of media, if you want to compare these two groups, you will use this format as the design formula. It's the same as linear regression. If you want to think about interaction effects, then you can do media multiplied by genotype. It's up to you what design you use, but a more complicated design requires a larger sample size. And here you can check the model matrix of your design. This is helpful if you are trying to understand the interpretation of each parameter. For example, here, when I use media as the predictor, it's actually coding a binary variable that is 1 for these 3 samples, 0 for these next 4 samples. these ones are actually the ones that are from the YPD media group, because this factor variable has the first level as tissue culture media, the second level as this one. Automatically, it's going to treat this first level as the baseline level. So it's going to code 0 for these samples and 1 for these samples. So this is the coding of the variable for media. You will know for sure which one group is used as the reference level. And if you have interaction terms, it is even more meaningful to look at the model design matrix. So here, when I multiply media and genotype, I'm actually creating three predictors. One predictor is comparing the two media groups. One is comparing the two genotype groups, and one is comparing, it's actually coding for the interaction effect. So in the previous part of the workshop, I also had a lecture talking about how to interpret the interaction effect. So this column is going to be these two multiplied together, and the coefficient for this column will tell you if the effect of media is dependent on the genotype. So that's the interaction effect, if the effect of one thing is dependent on the other one. And in order to really get a sense of how each sample's expected expression works, you can use this design matrix to get the coefficient. You can plug in the coefficient. You can multiply the coefficient for this column by this number, coefficient for this column by this number, and then the third column. By adding them up, you will get the expected expression level for this one. So this design matrix helps you to obtain the mean expression level for each of the samples. Any questions? That's just how the design matrix works. If you want to construct a DE-seq object for DE-seq analysis, you just need 3 inputs: the count matrix, the metadata matrix, data frame, and you need to tell DE-seq what variables you're trying to test on. This is contained in the design. And after you get this DE-seq object, you can obtain the metadata using the call data function. Call data function will give you that metadata table. And if you use assay, this function will give you the count table. When you run DE-seq, you can directly run DE-seq function on this DE-seq object. But inside DE-seq, it's actually estimating the size factors that's accounting for the library size and estimating the over-dispersion parameter individually. So it's going to estimate the size factor and then dispersion parameter before it does the differential expression analysis. So here, for some people, they may want to use the size factor for some other purposes. For example, they want to use the size factor to normalize their data. Then you can use the estimate size factor function on the DE-seq object. It will add one more slot to the column data. This is, so your metadata table will have one more column that contains the size factor. That's the library size. That's an estimation of the library size. It's 1 per sample, and you can obtain that through this function. So this will allow you to use the size factor to for other purposes, or you can account for this in some people may account for this in their DE-seq run. And then you can, after you obtain the size factors, the DE-seq object will contain a normalized count matrix. The normalized count matrix is essentially the count of the original count divided by the size factors of each library. And this normalized count, you can use it for some so when you have other analysis that require your count to be normalized by the library size, for example, you have there are analysis that requires like RPKM, those kind of things, then the normalized count data here will achieve that goal. It will divide the estimated library size. So after this normalization, you will have each library that's roughly the same size. So the normalized count can be obtained by using DE-seq normalize equal to true, or you can manually just divide the raw count by the size factor of your DE-seq object. If you, and the normalized count, I forgot, the normalized count is also used for visualization. Suppose you have identified differentially expressed genes and you want to visually check how differentially expressed it is, then it's helpful to use the normalized count to create that dot plot where you see one group is more highly expressed than the other one. If you use the unnormalized count, it doesn't make sense. So this is getting the normalized count for each individual gene for manual inspection of each gene. This is getting the size factor done, and then we'll estimate the dispersion parameter. This dispersion parameter is for each gene. And for some of the genes, you may run into an NA, which means that there's mostly likely not enough read count in this gene. So this gene is too lowly expressed. It cannot, DE-seq cannot estimate a dispersion parameter for this gene, and you will not be able to obtain the differentially expressed result for this gene. To obtain the dispersion parameter, you first estimate it and then use dispersions to extract it. This will tell you the dispersion level of each gene. So after those steps, you can perform DE-seq. Right now, the DE-seq version will do those automatically for you. We are just going through those steps to let you know what these slots mean in the DE-seq output. So essentially you can just run DE-seq on the DE-seq object you construct from the count matrix and the design and the metadata. And to obtain the result, you will use the function results. It will have one column for the mean expression level, one column for the log2 fold change, which is the coefficient for your regression parameter, which is the estimate for regression parameter. And this is the standard error of the estimate. So it's quantifying how well this is estimated, how accurate it is. The test statistic is the fold change divided by the standard error. And the p-value is going to be the value you get by comparing this with normal distribution. But right now we're testing the differential expression for thousands of genes. If you are comparing thousands of genes, you are going to, even if all of the genes are not differentially expressed, you're going to run into some small p-values just by random. So that's why we're going to adjust for multiple comparison and obtain a column of adjusted p-values. We're going to cover this in detail in the next section. So most of the times you are going to look into the log2 fold change and the adjusted p-values. If you want to, so this result is comparing the media YPD versus the tissue culture media. Because the tissue culture media, this is starting with the T, it's alphabetically the first, so it's by default the reference level. If you are interested in getting the fold change the other way, you don't have to rerun DE-seq. You just need to specify that in the contrast option. So when you run results of the DE-seq object, you can say I want to look into the media variable, but this time I want this one to be compared to YPD media as the baseline. The baseline comes the second. Then you will have another table of results, and all of the signs are flipped this time. So if you look at the first 4 genes, how do we interpret those coefficients? They're all having a negative log fold change. And these fold changes are from the comparison of YPD versus tissue culture. How do we interpret, for example, the coefficient for the gene 5? So for this one, it's negative, so it's suggesting that which group is having a higher expression level? The second one has the higher, yeah. This one has a higher expression level, and by how much higher? It's going to be roughly 2 times. Because this is 1, you do it to the power. You can many of the times you want the most significant genes ranked to the top. You can do that by arranging the adjusted p-value, and now you have the smallest p-value on the top. Those have the most significant fold changes. I guess we'll take a break right now and come back later for the visualization and the multiple comparison. I try to add some more materials to this DE-seq part to include the interaction effects, because sometimes people are interested in modeling the interaction effect and obtain the coefficients for that. My current version of the node doesn't have it. I'm trying to get it updated. But the useful function is the function called result names. If you apply the result names to the DE-seq object after the differential expression, you will see how each coefficient name is stored, and you can extract that coefficient directly. We'll see if I can get it done before the next break. So after you perform the differential expression, there are 3 kinds of plots that are mostly commonly used. The first one is called the volcano plot. So the volcano plot is a scatter plot with the x-axis for the log2 fold change and the y-axis for the minus log 10 p-value. So for example, here, this point has a minus log 10 p-value of 200. That means the p-value, the adjusted p-value itself is 10 to the power of minus 200. That's super, super small. So the higher you go, the smaller the p-value is. And the log fold change on the 2 ends are the largest log2 fold changes. Most of the studies I've seen, they would choose a cutoff for the p-value and the cutoff for the two-fold change, and only focus on the ones that are both significant and have a large fold change. Some of them may have a significant p-value, but it's a small fold change. It depends on your study. Some people are not interested in those. So they want both the effect size to be large and the effect to be significant. In order to obtain the volcano plot, you will obtain the results. This is getting the results data frame, where you will have the columns for the log 2 fold change, the adjusted p-values. This is how you're going to manipulate the figures for your own preference. So here we're going to specify the x-axis and the y-axis. And then after this point, you can change the color of the point. You can ask the ggplot to assign different colors for different points based on different criteria. And you can choose the color scale, like the palette of your plot, and you can change the background from gray to white. All of the changes, all of the things you learn from ggplot can be applied to this plot. The most essential thing is this data frame from results. And another diagnosis plot is the MA plot. It plots the log 2 fold change against the mean normalized count. Because sometimes you may have a large fold change for a very lowly expressed gene, something like around the left side of the plot. You may not trust it that much because it's all low expressed genes, and a small fold change for a low expressed gene may actually not be very interesting. So this is also a plot where people can narrow down the genes they want to focus on after the differential expression loss. And this is just overall. For this specific data set, you are seeing a lot of differentially expressed genes. For some data set, you may only see a few or maybe none. And if you want to zoom into one specific gene, we're going to use the normalized count, because before normalization, different libraries have different size, and that will confuse the visualization. In order to get the normalized count, you can use the functions we learned earlier, where when you take the assay count, there's a normalized equal to true. So this is how you obtain the normalized count from the seq object. It will adjust for the size factor. And this count can be used to visualize the individual genes. And the functions here I'm providing, these are just giving you a simple way of plotting the dot plot. But essentially, you can write your own code. You can just get this count matrix, take out that column, and just plot that column against your variables of interest. That's not too complicated. This is making it looking complicated. I inherited these functions from a previous lecture. But these functions are pretty convenient because now you have a wrapper that plots a dot plot for a single factor and for two factors. So for example, these two genes, the 5 and 7, one has a negative coefficient and one has a positive coefficient. And for the first one with the negative coefficient, you can see that there's a higher expression in the reference group. And for the one with the positive coefficient, there's a lower expression in the reference group. So this confirms the direction of the effect that you see from the output. Some of the situations you may have a less prettier or consistent plot. You may have differential expression with 3 samples versus 3 samples and still see a significant result. That's something that you need to be very careful about. For low sample size, it's always important to check the dot plot. And if you want to color them by 2 different things, if you want to plot each media separately colored by genotype, you can use the function I gave you, or you can construct your own way of plotting it. Do you guys want me to explain the code here? Not really interested? Okay. So the dot plot, and then after the differential expression, we're going to talk about multiple adjustment. So what is the difference between this column of p-value and this column of adjusted p-value? That is a very important concept because we're doing differential expression among thousands of genes. That means we're doing hypothesis testing like thousands of times. And in each hypothesis testing, you are going to make an error, either the type 1 error or type 2 error. And the type 1 error is rejecting the null hypothesis, concluding that the gene is differential when it's not. The type 2 error is concluding that we don't have enough evidence to say it's differential, but in reality, it is differential. So whenever we conduct a hypothesis testing, there's a possibility of committing one of those errors. And your test will have a trade-off between the 2 types of errors. If you are very conservative in rejecting the null hypothesis, you're more likely to commit the type 2 error. If you're very liberal in rejecting the null hypothesis, then you will more likely commit the type 1 error. And when in the tradition in statistics is to make a decision such that your type 1 error is well controlled under this probability level, while you try to maximize power. Maximizing power is the same as minimizing the type 2 error. So this is the decision rule that we're going to go with. We keep the type 1 error under control while we increase the power. If you're trying to test 10,000 genes, even if all of them are not differential, then just by random, you will have 500 of them being rejected. Because you're trying to control the type 1 error under this level. Just out of random, there will be this level of, this proportion of null hypothesis being rejected. If you calculate the overall type 1 error, the probability of making no rejection when the truth, when all of the genes are null genes, are non-differentially expressed. Oh, this is to the power of 10,000, sorry. If you calculate the overall type 1 error, it's actually going to be 1. So the probability of rejecting any, the probability of rejecting any of the nulls when they're all null genes is going to be a close to 1 if you do so many tests. So when you test so many genes, your overall type 1 error is close to 1, and you don't want that to happen. So you need to make the rejection of each individual hypothesis a little harder. And that's the philosophy of adjustment for multiple comparison. So there are 2 ways of adjustment for multiple comparison. The first solution is that we're going to control the family-wise error rate. Instead of trying to control the type 1 error for each individual hypothesis, each individual gene, we're going to control this thing called family-wise error rate. That is the probability of making at least one type 1 error. This one is going to control for this overall type 1 error. So the probability of making any wrong rejection. And you can imagine that is going to be very, very conservative. You're trying to control the probability of making any mistakes among 10,000 comparisons, 10,000 hypothesis testing. The way to achieve this is to, instead of rejecting a null hypothesis when p-value is smaller than 0.05, you're going to divide that 0.05 by 10,000. And if you do that, then your overall type 1 error is still going to be below 0.05. But for each individual gene, you're going to make the rejection so much harder. You're dividing the 0.05 by 10,000, the number of genes you're comparing. So this is a very conservative approach. The second solution is that we're going to control for the false discovery rate. This is the expected proportion of wrong rejection. So among all of the rejections, I'm going to make some mistakes. I'm going to allow for that, but I want to control the proportion of wrong rejections among all rejections. I'm not trying to control the rejection for each individual one. I'm just trying to make sure that overall the proportion of wrong rejection is under control, is below 0.05. And this is also called the FDR, is actually the one used for DE6. There are two procedures commonly used. The first one is the most common. It's called Bernardini-Hochberg method. It's assuming all of the genes to be independent. We know the genes are not independent, but it's hard to really obtain the correlation structure among the genes. So people they just blind themselves and just use that method. It's just easier. And the second one allows for positive dependence across the genes. But if you use this one, it's slightly more conservative than the Hochberg method. Most of the software packages you see out there will use the BH method to control for FDR. Questions?
How did you choose 1 and 2?
1 and 2? Solution 1 and 2?
Yeah.
1 is super duper conservative. It's rarely used. But you can see that Bernardini-Hochberg is assuming independence among all of the genes. So if you think you have a lot of dependency across the genes and you want to act more conservatively, you can use this method. Right?
Yeah. No, I like to think it's like false discovery rate. Like it's very much hypothesis generated.
Right.
Like you're like getting a lot of candidates for the future studies. If you were to compare the P and the values between those two, would they be within?
This one is almost always more conservative.
What is that? Can you?
What does it mean by conservative? Yeah. So this one will make your rejection much, much harder. Your adjusted p-value for this one will be much bigger than this one. So for example, this one is essentially, you're rejecting by, it's essentially multiplying your p-value by the number of genes. If your p-value is, if your p-value is 0.001, multiplied by 10,000 is already over 1, so it's going to be cut at once, and it's not going to be rejected. For this one, the adjustment is not straightforward. You cannot just multiply your p-value by something. You are more likely to end up with a smaller p-value with this method. I guess if you end up with a lot of differentially expressed genes, you can afford to be more conservative by using the first one. You can use either one, but it's important to specify in your manuscript like which method you use to adjust for the p-value. By default, DE6 will give you the second one. So if you want to choose the first one, you need to manually do the p-value adjustment. There's a function called p.adjust. You can input the list of p-values in there. It will spit out the adjusted p-value. And the p.adjust has several options. You can try to look at the help page. And here, it's too tiny for you guys to see, but you can see the BH, the BY, and the Bonferroni. Those three are the ones that I covered in the lecture. FDR is the same as BH, is just another name. Which one? You like home?
It's Bonferroni. The control of the family-wise, but it's a little less conservative. So it's like the first will be 0.05 divided by the number of tests. That's significant. It's 0.05 divided by the number of tests minus 1. So like the threshold gets a little less each time as the p-value has to cross. But it's still also much more conservative.
Yeah, because when you have 10,000 minus 1, then it makes sense. Yeah. But
there are a way of basically checking whether these two methods are calling what is significant. I guess.
Are you asking whether we can check the consistency between those two?
Well, not so much the consistency, but for the accuracy. That if I'm calling expression of a certain gene significant, that like the differential expression significant between groups, it is actually significant, not just
a false.
Yeah.
There's no way to know. Yeah, nobody knows the underlying issue. But a good practice is to, if you're really into that, you can use several different methods. Like you can use RNA-seq and you can use lemma. They both call this one as differential gene, then it's more likely that it is. Yeah. But for the correction, I believe that most of the correction, the adjustment for p-values, they're mostly monotone. Like if you have a higher p-value for this gene than that gene, then after adjustment, they will maintain that order.
For positive dependence, does that mean that the expression of one positively influences the other?
They tend to be more expressed together. Yeah.
So so if your data set has if you're looking at a bulk sample of I'm trying to figure out a reason to use the BY.
Actually this is positive dependence. I think is they tend to be rejected together in that. So it can be one is more positively expressed, the other is more negatively expressed. So they tend to be more significant together.
So it's like looking at kind of families of genes that typically.
Yeah, because you're adjusting the p-value. The p-value doesn't have a direction of the effect. Yeah. So after the multiple comparison, your adjustment p-value is always going to be slightly larger than your original p-value. And the volcano plot is created using an adjustment p-value. You'll always report genes based on the cutoff for the adjusted p-values. If you change the set of genes to test, your adjustment p-value will also be different. So that's the importance of filtering the really trash genes at the very beginning. Now you will end up with a smaller set of genes. You will suffer less from the adjustment to multiple comparison. So here we have example for the family-wise error control and the FDR. Suppose we have 20 genes, they're all independent. 10 of them are differentially expressed, 10 of them are null genes. And we have a test that has a type 1 error rate that's 0.05. So the probability of my test rejecting null genes will just be 0.05. And my probability of rejecting the DE genes, so that's correctly identifying the DE genes, it's going to be 0.8. This is my power. So for a test with this type 1 error and this power, what would be the family-wise error rate and FDR? So the family-wise error is the probability of making no wrong rejection. That means that for all of the 10 genes, 10 null genes, I cannot make any rejection. And that probability is going to be 0.4. For the false discovery rate, this is the probability. This is the number of genes I'm going to reject wrongly. And this is the total number of rejections I'm expected to make. FDR is the proportion of wrong rejections among all rejections. This is wrong rejection. Among all of the 10 genes, I have 5% probability of rejecting. This is the total number of wrong rejections. This is the total number of wrong rejections. This is the total number of right rejections. And if you calculate that, the FDR is this number. So when I'm testing among 20 genes, my FDR is actually not inflated by much. My test is controlling type 1 error at 0.05. My FDR is not too much more than 0.05. But my family-wise error rate is much larger. So if you want to press this under 0.05, you will need to adjust more on the p-value. But if you want to press this under 0.05, you just need to adjust. So that's why this is another example showing this one being more conservative than FDR. Any questions? So this multiple comparison is going to be used in RNA-seq. It's also going to be used for gene set enrichment analysis, because in gene set enrichment, you are going to test multiple gene sets, and that will also need to be adjusted for. The more gene sets you test for, the more adjustments you're going to make. If you're only focused on a few gene sets, you don't have to run the whole database of gene sets. So I think it's very important for you to clean up your gene set before doing the gene set analysis. So we'll go over the gene set enrichment analysis. There are many methods for gene set enrichment analysis, and we're going to go through the most traditional one. This one is, I believe, the philosophy is the most straightforward. Some other ones require other assumptions and have a lot of manipulations inside, but this one is more straightforward. The GSEA method identifies gene sets that are overly represented. So if you have a set of genes ranked, if you rank all of the genes from top to bottom, depending on whatever criteria you have, maybe depending on the log two-fold change, or depending on the p-value, the adjusted p-value. You rank them from the top to the bottom, and some of them will belong to your gene set, some of them will not. And you're trying to see if your gene set is located on the top or bottom or everywhere in the list of the genes. If your gene set is more represented by the top genes, you have a positive enrichment score. If your gene set is more represented by the bottom genes, you have a negative enrichment score. If your gene set tend to be spread out along the list of the genes, then you have a not significant gene set enrichment score. So that's a philosophy of the overrepresentation. You want to check for each gene set if this set of genes is overly represented by the top or the bottom ranked genes. So the input is going to be a list of genes, gene sets. You can have one gene set, or you can have multiple gene sets. And you are going to have a vector of gene level statistics. They can be the test statistic in RNA-seq output. They can be the log2 fold changes. They can be the minus log 10 p-value multiplied by the signs of fold change. So this is going to be the larger, the more differentially expressed it is. It's going to be roughly in the same order as the test statistics. The test statistic and the minus log 10 p-value multiplied by sign, those two account for both the fold change, the effect size, and the significance level. Because this one test, it will be the fold change divided by the standard error. So it counts for the fold change and the significance level. This thing is the same. It's a transformation of the test statistic. The log2 fold change doesn't account for the significance level. If you use that as the enrichment analysis, I've seen that a lot, but it will not, you will likely run into some genes that are ranked on top, but they actually are not significant. So it's up to you what you use for the enrichment analysis. I prefer to use the first two. And you need to put all of the genes you run RNA-seq on. You cannot just throw in the significant ones. This is a very common mistake. You need to include both the significant and non-significant ones. And the good side of this is that you don't have to choose a cutoff for the adjustment p-value. For some other enrichment methods, you will have to choose a cutoff, for example, 0.05 for the adjustment p-value, and only input and tell the enrichment method which genes are DE, which genes are not. But for this method, you don't have to provide that cutoff. So you provide the gene sets as a list. You provide the vector of gene level statistics for all of the genes. And the software itself is going to rank the genes by their gene level statistics. And then it will calculate the gene enrichment score. So I have like large chunks of text describing it, but I think it's just easier to look at this figure from their the GSEA paper. So you have all of the genes expressed. Each row is one gene, each column is one sample, and your samples are divided into treatment group and B. And the genes are going to be ranked based on their gene level statistics. The top ones, you can see that they're more expressed in A. The bottom ones, they're more expressed in B. So the genes are ranked by their statistic, the gene level statistic. And this gene set is, the bar means that the corresponding gene is in this gene set. The blank means it's not in the gene set. So this is indicating all of the genes in this gene set. And this is turning it around to horizontal that way. This bar, the length of the bar is the value of your test, the gene level statistics. So your input is going to be this, and the gene set. And how do we calculate the enrichment score? We're going to have a random walk where you start with 0, check each of the genes from the top to the bottom of the list. If this gene is in the gene set, you walk to the positive, you walk up. If the gene is not in the gene set, you walk down. So each gene, you take one step, depending on whether it's in the gene set or not, you walk up or down. And your step size is dependent on this gene level statistic. The larger your, for example, if we're using the test statistic as the input, then the larger the test statistic is the larger the step is going to be. So it's going to do a random walk by going through all of the genes from the top of the list to the bottom of the list. Going up when the gene is in the gene set, going down when the gene is not in the gene set. If all of your genes in the gene set are in the top of the list, what will happen? Suppose that all of the genes are here. There's no bars there. They're all in the top of the list. You're essentially going to walk up a lot in the very beginning, and those are big steps. And when it comes to the small steps, you will walk down, walk down, walk down. So you will have a bump at very early of this random walk. And this distance between the peak of your random walk to 0, this is going to be the enrichment score. Yeah.
So that statistic in the middle, the correlation.
Yeah. Yeah.
That's how is that determined?
That's your input. That's the gene level statistic. If you're using log 2 fold change, it will be log 2 fold change. It's quantifying how strong the connection is between your gene and the gene set.
That doesn't then become negative. This is a negative step later on.
It does. So when it's negative, it's the step size is dependent on the size of this.
They don't step down, they step up just less often the genes are right?
They step down when you run into a gene that's not in the gene set, so they do step down here.
Right, but a gene, this is a gene set that's on, right? It doesn't have a negative effect on this. It doesn't like step them down, still steps up just less than the gene. Alright. I didn't know about the 6 thing that changes the size of the step. I thought it was literally just based on ranking the genes and from there getting an effect.
So when you're ranking the genes, the top and the bottom, they're both DE genes. It's just the top are the positive DE genes and the top are the upregulated genes and the bottom are the downregulated genes. Yeah. So at the beginning and the end of the random walk, you will both take very big steps, and in the middle, it's the small steps.
So the enrichment, is it the length of the peak up to the peak? Or
it's not the horizontal distance, it's the vertical. It's how far you can walk away from 0.
Okay.
What is the range of that score?
Well, it can be very big. It depends on the number of genes, and then it also depends on the effect, the differential expression effect. So that's a good question because you don't really know how large it is to be large. That's why there will be a normalized enrichment score. The normalization is done by scaling it in comparison to a random set of genes with the same size. So suppose this gene set has 40 genes. Then I'm going to randomly create a gene set of 40 size, like 40 genes in there, and calculate the enrichment score for this random set of 40 genes. And I will do it repeatedly to obtain like a null distribution of the enrichment score. And that can help me get the scaling of the enrichment score. Generally, when you have a larger gene set, you can afford to walk for more, right? If you only have 1 gene, 5 genes in the gene set, you can only walk up 5 steps. Any other questions?
To rank the genes, they use
the input gene. You can use fold change or test statistics or log 10 p-value.
And that those data will affect the weight of each step, the also based on.
Yes, the step size depend on this. So if your gene set is spread everywhere, then your chance of walking up and down are going to be evenly spread. You will likely run into an enrichment, like a random walk that's like this shape. And if you have a peak on the bottom and peak on the top, you're going to choose the largest one. If it's on the top, and then it's positive score. If it's on the bottom, it's a negative score. If your gene sets, for example, are all located at the bottom of the list, you will walk down first and then walk up later. And that distance is going to be your enrichment score.
It's like the normalization applied to the enrichment scores?
So the larger gene set you have, the more enrichment score you're going to have, right? So we normalize that by the, right now I have this gene set that is meaningful, and I'm going to create gene sets that are not meaningful, just randomly choosing 40 genes out of all genes. And for each random set of 40 genes, I will calculate that enrichment score, and I will take an average of them.
And then you kind of like subtract.
Divided it. Yeah. But that doesn't give you the significance yet. It's just a normalized enrichment score. To get the significance, you will do something like permutation of the gene labels as well. It's similar as the random set of genes, but this time you're going to randomly assign the gene sets membership. You're going to shuffle the gene labels and calculate the same thing. And that will give you a null distribution, and you will see the position of your observed enrichment score in that null distribution. And the GSEA package, FGSEA package will also give you something called the leading edge gene set. So that is the set of genes. If you're having a positive enrichment score, that is the set of genes right before the peak. So for this example, all of the genes on the left of this line are going to be outputted as leading edge genes. If you have a negative one, it's just right below, behind that line, the peak. So the description here is for you to understand afterward if you want to review the lecture. But the concept is all described in this figure. To implement, we are going to use the test statistic from RNA-seq. And you need to make sure that the test statistic numbers each correspond to one gene name. So if you just do this line, it will give you a vector of numbers without knowing which one of them is which gene. Now we're going to assign the names of the genes to each number. This will be used as the gene level statistics. And for pathways, you can rely on multiple databases. The GSEA website provided this set of gene set annotations. So there's Hallmark, there's several curated gene sets, and there's other databases like GO or CAG, a lot of like annotations out there. If you're interested, you can actually combine multiple databases together. But the more gene sets you are testing, the more likely you're going to run into like redundant gene sets. Some gene sets are just named differently, but they're actually the same thing. So if you're running a lot of gene sets, it's important to do some cleanup of the gene sets. But if you, for example, if you want to use a Hallmark gene set, it has 50 gene sets, then you will download the GAMT files, either by gene symbols or this gene IDs. There are multiple gene naming systems. They should have matching, like there are softwares that can help you match the gene name. for some data sets, they may store the gene names using this gene ID. Some will use gene symbols. It's like a pretty chaotic, but there are functions to help you match the gene names. You need to make sure your pathways list your gene sets have the same naming fashion as your gene specifics. So if you, after you download the GMT files, you can transform them into a list of genes through this function. It will convert the GMT file into a list, an R list, and each member of the list is one gene set. It's going to be a vector of genes. So that's how the gene sets are stored. And you can also use these functions to map gene names from different naming fashions. These are the two popular ones. It's quite the case by case, so you probably need to Google a little bit. For the illustration purposes here, I'm going to manually define 3 gene sets. So I just chose some of them. It's not meaningful, but this is how you can specify gene sets yourself. So I have collaborators who are just interested in certain functions of genes, and he's going to give me a gene set that's customized, so I can input them in the enrichment analysis myself. You don't have to always rely on databases. So the 3 gene sets I have here are stored as a list. Each list member has a name for the gene set, and the element of each element is a vector of the gene names. So this is the gene set, and I have the gene level statistics. I'm ready to do the enrichment analysis by using both as the input. And you can specify for gene sets smaller than 5 genes, I'm not going to do any enrichment analysis. For gene sets that are more than 500 genes, I'm not going to do any enrichment analysis. Why bigger gene sets are still not analyzed? Because when you have super large gene sets and not too many genes, then it will be harder to calculate the null distribution by shuffling the genes enough. So your gene set is already taking a large chunk in the background. By shuffling the genes, you're not getting a very good estimation of the null. So these can be specified. And also, like for super large gene sets, it's not super meaningful to look at their enrichment anyway. And this unperm option, if you don't specify it, it will run a different version of GSEA. It's called the something like multi-level FGSEA. So it's not the original version. It is using a slightly different philosophy as what I described earlier. If you want to use this most old-fashioned traditional one, you need to specify the unperm here, and it will also specify how much resolution you want on the p-value. So when you specify something like this, the smallest p-value you are going to get is going to be 1 over this number. The more digits you want in your p-value, the larger number you want here. But it's also going to be slower if you have a larger. This is the number of permutations you do to shuffle the gene labels to obtain that p-value, the permutation p-value. So after when you run this, it's a good habit to set the seed because it's a random permutation test. So if you run it a different round, it may change slightly. And then the result is going to be a data frame. You will have each pathway name, the corresponding p-value and adjusted p-value, the enrichment score, the normalized enrichment score, and the leading edge genes are stored in this whole string. This is the size of the genes. This is the number of more extreme genes than this gene set, but I rarely use this column. Most of the times you will just extract this column and this column, the adjusted p-value and the normalized enrichment score. You may make a heat map with rows ranked by the enrichment score and colored by the p-value, something like that. Or you can make a volcano plot with the enrichment score and the adjusted p-value. The leading edge gene set can be pretty large, but for some of the gene sets when it's small, it's good to see what are the genes that are driving the enrichment of that set. To plot so GSEA has a built-in function to plot the random walk of your gene set. So for my third gene set, I have about 200 genes. This is the random walk it's taking. Because I just randomly choose the genes, you can see the random walk is like up and bottom is like an S shape. So the enrichment score is actually pretty not significant. If it's more significant, it will probably go like double the size at the beginning and then go to 0. So that's all I have for today. I think next year when I teach it, I will probably run through the code together with you guys in the lecture so that I make sure you can run the code by yourself. But I think if you want to run the code now here, I can stay here and help you. Yeah.
Well, I wanted to know whether, how can you tell, you mentioned that you can see that it is not significant. How can you tell visually without looking at the p-value?
So the gene sets, the black bars are everywhere, like it's evenly spread across the background. If it's more concentrated at the beginning, then it's, yeah.
And if it was concentrated at the end, it'll drop at the beginning and then go up.
Yeah.
So there's also this, gene set enrichment analysis software. It's pretty popular. I know where you like import, whatever file that way. And now other people will use CFGS package in R. Do you have any opinions on like the? Do you know what software I'm talking about?
Which one? What is it called?
It's called. I was called GSEA, Broad Institute, GSEA software.
I think it's the same. This is the same implementation, just in R.
Okay. Do you think that's it?
There's slightly difference in the original implementation and this one. This one takes the gene level statistics, but the super original GSEA actually takes in the expression matrix. And when it's calculating the p-value, it's not shuffling the gene labels, it's actually shuffling the phenotype labels, like treated versus untreated in the expression data set, and do the gene expression DE-seq again, and then calculate the enrichment score for that shuffled data set. So it's actually shuffling the samples in the genes.
Yeah, so the software has the different permutation types got two different types of permutations.
Yeah.
Type for the genes.
Yeah.
I believe for you have fewer samples than like 7 or so, they recommend you
shuffle the genes
that permutation. Right? There, I guess it's probably.
For this one, there's no way to shuffle the samples because you are not telling the software what phenotype labels each sample corresponds to.
This one already does the genes that permutation.
This one permutes the genes, yes.
Okay.
It's like the one that you mentioned that shuffle the gene labels, the phenotype labels. That one is more, I would say, more solid, but it's just slower and more sample size. Yeah. And there are also other enrichment methods that require you to choose a cutoff for DE versus non-DE. And it's basically getting a 2x2 table, like DE, non-DE, DE in the gene set, DE, non-DE in the gene set, DE not in the gene set, and non-DE not in the gene set, and try to see if that is significant.
Could you remind me what kind of statistics was used for that, for this data?
Yeah.
Was that correlation?
No, so this correlation with phenotype is actually your input, that is the output of DE-seq, is your test statistic or log 10 p-value.
Oh, okay.
Yeah. You rank the genes by the significance or the log 2 fold change.
But if you use just the log 2 and not the p-value of the test statistic, would it be meaningful still, like to do the random? I mean, it wouldn't.
Some people would do that. It's a different interpretation. You're trying to see if your gene set is overly represented by the most DE genes, but you won't be able to say whether they're upregulated or downregulated.
Okay. Yeah, I'm asking that because I have a data set. I did like some log sequencing that I didn't run any replicates, but I was just curious if I could follow what this is. You should DE-seq.
You don't have replicates for
my sample. So basically, ran one sample for condition instead of like 2 or 4 like you had here.
Oh, yeah. I don't think you are able to run DE-seq.
Yeah, I mean, I looked into it. I think that's something. If you don't have it, you cannot.
Yeah, you don't have enough degree of freedom to estimate the parameters.
Oh yeah, because you'd be like calculating, like shrinking that based off of like a single data, right? Rather than a distribution between multiple data. Right?
Yeah. So you have like the rule of thumb is to have more samples than the number of parameters. In this example, we're having treatment as one parameter, right? The overdispersion, that's one parameter. So at least you will need to have enough sample size for those two. So that's going to be 3 samples. And for DE-seq, you're testing multiple genes together, so that makes it more complicated on the sample size calculation. But I barely trust any results that's 3 versus 3. Like 3 samples versus 3 samples, unless you have a really strong effect that all 3 samples in one group are expressed, all 3 samples in the other group are non-expressed at all. Those kind of things I can trust. But like 1 non-expressed versus 2 non-expressed, that's just too much.
So this was just a pilot run, if you know.
Yeah.
Genes, like I had like 2,000 sequence.
Yeah.
I had full critical permission on it. It was doing a real experiment with proper control. I was just curious. If just looking at the whole chain would give me some information that, you know.
In your case, DE-seq won't even run. So the fold change you have is going to be more like you can manually calculate that by taking ratio.
That's what I want to try to come up with.
Yeah. So any questions about the other part of the lecture?
I have a question about like Go enrichment. Do you similarly not filter your significant genes if you were to use kind of like a pathway kind of analysis using Go terms and stuff? Would you similarly like not filter and just use all of your genes? Or you would presumably filter to just your significant genes?
So the filter part is related to what enrichment method you use. The Go thing is related to the database for the pathways. So you can actually use the Go database to format your list of gene sets, but still use the FGSEA for enrichment analysis.
Okay, so you can feed in like Go
whatever, yeah.
And then run effectively the same code.
Yeah, yeah.
Rather than getting gene sets to get
Mm-hmm. I think I always go too fast. I feel like I prepare a lot of materials, and every time I go too fast.
You're good. You're good. Actually, keeping it short.
It's exhausting for you guys to sit there. We need to talk about shortening the session.
Thank you.
Thank you for coming. Oh, the survey, end of the lecture. So there's a survey for the lectures. We can make it better for next year.